Hello and welcome to a podcast from Odell Technology. Today we're talking to Dr. Nikolas Pontikos, who leads the UCL Institute of Ophthalmology on combining retinal imaging, particularly fundus photography and possibly OCT, with whole exome and genome sequencing data and phenotype annotations. Hello, Nikolas.
Hello, Stephen. Thank you for inviting me.
Okay, it's a pleasure to have you here. This AI is trained to recognize distinct visual markers in the retina and correlate with known inherited retinal diseases. And I think it's a very impressive technology. I was wondering if you wouldn't mind telling the audience a little bit about yourself, please.
Yes, of course. I'm an associate. I'm currently an associate professor at the University of College London, and also a co-founder of ITGene. My background's in computer science. That's nearly 20 years ago now, and I've from computer science I moved into bioinformatics, working with genetics data. And I was working on a range of different diseases and conditions, including Crohn's disease, type 1 diabetes. And I found my way into ophthalmology and working on rare inherited retinal diseases on the genetic side. Um, and this was around in 2015. And working in that space, just the potential of AI and of the data sets that are available in ophthalmology with regards to fundus imaging, retinal scans, and so on. That opportunity that opportunity really excited me to develop some AI system. And this was after talking to doctors who have expertise in these inherited retinal conditions, but recognize that that expertise is not democratized to many places in the world. That led me to develop the ITGene project nearly, I would say, six or 7 years ago, and we've been working on this now for a long time, steadily through grant funding, and we've recently published a paper on this now, and yeah, we're very excited about the next steps as we think this technology has a lot of potential.
Oh, well, congratulations. Many IRDs have overlapping phenotypes. How does eye to gene distinguish between, say, Stargardt's disease and cone-rod dystrophy based on fundus imaging?
Yes, so you're absolutely right, Stephen, to highlight that there's a lot of overlap in the phenotypes, and there's sometimes a lot of diversity even within one gene in the phenotype. So of course this is challenging, and this is the reason why human experts, have a hard time identifying genes from retinal scans. Um, but ITGene is able to circumvent this by through the volume of data it's able to process, and in order to disentangle and dissect these gene phenotype correlations. So we do this in two ways. We do this in training a classification algorithm, so a convolutional neural network, and actually it's an ensemble of convolutional neural networks which are trained on three different types of imaging modalities: fundus autofluorescence, OCT, and infrared. And then the second way we approach this is by using a clustering approach, which is based on the representation of the images as they go through this neural network. That representation is a high dimensional space which gets projected onto a two-dimensional space known as a UMAP, and then we apply clustering on that in order to identify retinal scans which have a more similar phenotype, and from that uh, infer ones which have a similar gene condition. Um, but it's still work in progress, and sometimes, as you pointed out, phenotypes can be very difficult to disentangle.
Absolutely. Are there specific retinal regions or markers that the model weighs more heavily on? And are those consistent with what human retinal specialists focus on?
Yes, so using the first approach, which I mentioned, the classification network, we're able to then uh, apply what's known as attention maps to this in order to identify those regions which are highlighted as being important, in the decision process of distinguishing the different genes. And these we found do seem to overlap with what human experts would use. And then another approach that we are publishing, we have recently published a paper on, and we have another one that is being submitted, is to actually segment out the features that we think are relevant, and then to use that as the basis of distinguishing the different genes. And this work that we've published in Ophthalmology Science shows that we are able to recapitulate a lot of the known gene phenotype associations, as well as discover novel ones.
Okay, so are you using multimodal learning? You're combining the fundus with OCT and clinical notes and genetic variants?
We are not combining genetic notes yet, but it is our intention to extract information from the clinical notes. We are already using the mode of inheritance because that is important, and the age of the patient, because that can also be useful in distinguishing the different genes based on the age of onset and whether the disease is progressive or not. But at the moment, we are using multimodal imaging, so different types of imaging modalities.
Okay, and I imagine you employ saliency maps or GradCam to explain model predictions.
That's right.
Yes. Okay.
Exactly. So those are the attention maps I mentioned yesterday. It's also known as you said, saliency maps or GradCam.
Okay, that makes perfect sense. How does the system handle novel variations of genes of uncertain significance in terms of diagnostic confidence?
So the variants of uncertain significance that so I guess that maybe I'll split my answer in two. So there can be variants of an uncertain significance, and those are usually in genes which are known to be associated with inherited retinal diseases, or of which there are over 270 genes which are known to be associated. Those variants genetic variants usually are reported from an exome or whole genome, and they have evidence which indicates that they may be pathogenic, but then not sufficient evidence to really tell whether they're pathogenic or not. And so what RTGene adds is the information that comes from the phenotype to say how well the gene fits the phenotype. And that's a specific line of evidence that is very useful in inherited retinal diseases, and that is used all the time. And that line of evidence is usually coming from a clinician expert, which these experts have been looking at diseases for a very long time, and they only really exist in centers of specialization in these diseases because they are not commonly seen in the community. And so with ITGene, we're able to provide this evidence through the AI and to do it in a more quantitative way. So that can help in classifying these variants of uncertain significance as either pathogenic or not. And then the second is whether it's an unknown gene or a gene you have not seen before, which I think is another possibility. It has been trained as a classification tool on, say, up to 63 genes. But outside of the 63 genes, we have to resort to a clustering approach, which is what I mentioned previously, in order to identify retinal scans that may look the same, but we don't necessarily know what the gene is or if the gene phenotype fits. So using clustering, we can identify novel clusters, which may be novel, which then we can analyze the data of these patients, the sequencing data of these patients from whole exome and whole genome, and see whether they will find any common genetic variants in certain common genes, which may explain, which allow us to discover a new genetic cause of the disease.
Okay,
if that makes sense, yeah.
It does make sense. It's fantastic. Could the model eventually help identify new syndromatic associations or propose novel disease genes? Is that possible?
Yes. So using this approach, we could this clustering approach, we could identify patients that phenotypically look similar, and then that can power gene discovery. And even within Actually, what is really interesting is within a specific gene, depending on what part of the gene is affected and what type of mutation, genetic variant occurs in the gene, the phenotypes can look different. And so we are able to quantify that now in a way that we were not able to do before. So we can show, for example, if somebody has a genetic variant in this specific location, the phenotype is expected to be more severe, for example, or faster progressing, and so on.
Okay. So are clinicians already using ITGene in exploratory mode?
Yes, so we are so we have a lot of static sort of results that we've run on retrospective data, and we are sharing this with the clinicians and this giving them some novel insights. In terms of deployment internal, we are also using it as a research tool at the moment in education sessions, for example, with clinicians. The clinicians have weekly sessions where they look at retinal scans, and then a more experienced clinician will say what they think is the gene diagnosis, for example, and then it will be revealed later in the workshop. So we are using ITGene in that setting as well as an educational tool. For it to be deployed in clinical practice, obviously, it would require regulatory approval, but it can be used as a research tool for now, in particular
Okay.
for variant prioritization.
So in a head-to-head comparison, how does ITGene perform versus expert genetic ophthalmologists on very complex or ambiguous cases?
Yeah, so overall, we did a benchmark which we included in the paper published in Nature Machine Intelligence last month,
Yes.
and we found that even for the clinicians who are the most experienced, ITGene is still outperforming them by a factor of twofold, or even 1.5 to twofold. So it's doing much better. And I think that's to be expected because it's just very difficult problem for clinicians to do. And unless we've been looking at many of these patients, or they actually recognize the patient from the retinal scan, it will be very difficult for them to identify the gene in a lot of the cases. They will be able to tell some of these sort of easier cases which have a very distinct phenotype. Um, for example, Best disease has a very distinct phenotype, or retinoschisis, choroideremia, but most of them have a more ambiguous phenotypes, which are very difficult to tease apart.
Okay, so I think you've maybe answered this, but I think given the differences in allele frequencies and fundus pigmentation, what steps have you taken to ensure ITGene performs equitably across different ethnic variants and across different geographic regions of the world?
Mm-hmm. Yeah, so sorry, I haven't touched upon the external validation yet of ITGene, which is also something we've done as part of that recent publication.
Yeah,
So we have validated it on data on retinal scans uh, from other European countries, but also from more ethnically distinct populations, like from Tokyo, Japan, and from Sao Paulo.
okay. Oh, that's interesting.
And we
Okay.
found that the performance is fairly consistent across the ethnicities, even though the gene distributions, as you said, the allele frequencies or
Yes,
the gene prevalence are different in different countries, in particular those in, for example, in Japan and in Brazil, they would have
sure.
different prevalences there.
Absolutely. So how do you see ITGene reshaping ophthalmology and clinical genetics training? Should the next generation of clinicians learn to interpret fundus AI outputs just as they learn to read CTs or the ECGs?
Yeah, that's a really great question. It's a bit early to say, but I certainly see it as a tool um, for education, in a way to make it more interactive. I think for when
Yes.
clinicians are trying to interpret retinal scans, um, and to identify the likely gene. Um, I think it's it's going to bring more attention to these diseases, which is a good thing in places where diseases are neglected or misdiagnosed. So that's going to be, I think that's going to make a big difference in countries which do not have access to genetic testing, and where these patients are basically not managed at all. They're just told they have something wrong with their retina, probably genetic inherited, but that's about it. That stops there. So
Yes.
having all that extra information there is going to be very important, and it's also going to allow them to make a link, hopefully, to experts. So it's going to be at the same time a telemedicine opportunity, so that it can link patients in these countries, for example, to experts in other countries, or to genetic testing companies who would, or pharma companies, more probably even more relevant, who
Yes.
are looking
More
for these
relevant.
patients. Yeah. So I think it's going to have a huge impact in the places where genetic diseases, inherited retinal diseases, are just not managed. Is I think within these centers of specialism, like the ones, for example, we have in the UK, like at Malthus Eye Hospital, it's going to be useful, but it's going to be more maybe for educational purposes because the expertise is here. But hopefully, it's going to enhance the level of expertise as well of the clinicians in ways which are, as you said, more quantitative, looking at interpreting the outputs of the retinal scans rather than purely just pattern association. So in a way that we can say, for example, based on this combination of parameters, we think it's this, and we that was also going to give us more understanding of how diseases progress, which at the moment is quite qualitative. Actually, a lot of the data is quite qualitative in this space. It's not very quantitative, so we're
I
trying to
didn't
make
know
it more
that. I
quantitative.
didn't know
Yeah.
that. Okay.
Mm.
All right. Oh, that's interesting. Well, that's a game changer for pharma, isn't it?
Yeah.
All right. So tell me, is there something that you'd really like to discuss that you're passionate about regarding this technology that we haven't covered?
Yeah, I'm really passionate about seeing it used in countries where patients just do not have access to clinicians who are able to diagnose these diseases because they don't have access to genetic testing. I mean, for me, it would be immensely rewarding to see that, patients in these countries now all of a sudden have access to information and support, and they are now visible as well to these pharma companies. Um, I guess what hopefully will derive from that is that this in turn will stimulate more uh, investment into pharma, and provide more incentives for pharma to
Yes.
develop treatments for these patients if they know where they are in the world and who they can, and if they can recruit them. Um, and we can
Yes.
help them optimize their trials as well by finding the right patients for them. We talk a lot about precision medicine, but I think the gene therapy, the gene targeted treatments that have been developed are really already precision therapy that's with us today. It's just now a problem of identifying these patients and supporting the clinical trials by helping them with recruitment of these patients and finding the right patients for the right trials. So these are things I'm very passionate about, making a difference. Um, for these patients, in particular the ones that are very isolated because they just don't have access to
Nicholas, I
any
think without
kind of
knowing it,
support. Yeah,
you're actually going to affect the lives of millions and millions of people.
I
I
hope
think
so.
that's what you're going to do. I'm sure you are. You'll have a huge impact
Mm.
on the health of millions of people. So I wanted to thank you very much for your time today in engaging with us. I hope to talk to you again in a few more months, 6 months, and see where we are with the project. But thank you very much for your time and your research. Thank you very much.
Thank you so much, Stephen.